Welcome to the third year of the CRC Roundtable, our monthly virtual seminar series. Just as a reminder, our intention is to host these targeted, inclusive, and informed conversations that match scientific advances and management needs. The webinars are designed for contribution, not consumption, and as a reflection of that intent, the panelists have been given about 25 minutes total to make some introductory remarks, allowing 30 minutes for discussion. This is all about us, giving us a place to ask the clumsy questions that we've been waiting to ask. Let me start, as we always do, by understanding why this topic at this time by these speakers. Today, we actually continue our exploration of per and polyfluoroalkyl substances, which I will call PFAS from now on. Today, we actually began that journey in December of 2021. PFAS have been manufactured and used in a variety of industries in the US since the 40s. They're ubiquitous and persistent in the environment and have the potential to have adverse human and ecological health effects. The potential risk is significant. EPA published interim health advisories in June 2022 in recognition of new science that indicated that some negative health effects may occur with concentrations in water that are near zero. Why this time? As a reflection of the speed at which we are realizing the consequences of PFAS manufacture and use, on March 14th, last month, EPA released a proposed national primary drinking water regulation for PFOA and PFAS. PFAS, I just learned this talking to our speakers, is the first non-carcinogen ever regulated by EPA in the States. And so this is certainly new ground. However, while we're recognizing the risk at breakneck speed, we still do not know much about the sources, distribution, and effects on fish and wildlife, all critical uncertainties as we assess the danger that this poses to our restoration efforts. And also note that there are more than 12,000 unique compounds in this class, making analysis and reporting difficult. Why these speakers? They represent, as you'll come to find out, both the production of new knowledge and its implementation into the existing effort. So that's a complete arc that is illustrative of the importance and challenges of adapting a science-based restoration program in a changing world. I'm really excited about our speakers today. Kelly Smalling is a research hydrologist with the USGS. She's was co-chair of a STAC workshop steering committee, along with Michelle Laura, on PFAS. She's an environmental organic chemist who has been working with the USGS since 2004. Her research focuses on the occurrence, fate, and effects of emerging contaminants, including current use pesticides. But she also loves amphibians, and she's getting ready for Amphibian Week, May 7th, I believe, through the 13th. So she's getting pretty excited about that. Emily is a hydrologist, again with USGS, and she's been vice chair of the Toxic Contaminant Work Group, or she is vice chair of the Toxic Contaminant Work Group. The Toxic Contaminant Work Group has a really big task. It was established in 2014, and I'll just note that it tries to keep an eye on the over 80,000 industrial chemicals and their potential effects, many of which are invisible. Emily has a wealth of experience in investigating the nature and extent of contamination in groundwater, soil, surface water, wetland environments, and indoor air. And what she's working on in the moment isn't as exciting as Amphibian Week, but she is looking at PCBs in stormwater in non-tidal wetlands around Baltimore. They had differing views of what they wanted the weather to be this weekend. Kelly is trying to run a race. She did not want it to rain. Emily wanted it to rain so she could get a stormwater event. So with that, please note that we will facilitate the discussion out of the chat, and I will do that. And at this point in time, I am going to turn over the floor to Kelly and Emily.
All right, thank you, Denise. I'm going to share my screen and get this going. Can everybody see this?
Looks
Yes.
good.
Excellent. So let me start my video. I am very excited to be here today, and thank you, Denise, for a wonderful introduction. You actually probably took about 3 minutes off my presentation by covering some of the basic information about PFAS. So I can breeze through some of my introductory slides. But I first want to thank everyone for joining us today, and I'm really looking forward to a really great discussion after Emily and I give you all a brief overview. As Denise mentioned, I'm with the U.S. Geological Survey. Both Emily and I are. I'm stationed in New Jersey, and I've been working on contaminants in the Chesapeake Bay for almost a decade. For those of you that aren't familiar with the USGS, we're the nation's premier earth science agency. We do not regulate and we do not make policy, but we do try to provide timely science and defensible data to other federal and state agencies that do make policy and do regulate. As Denise mentioned, we're going to talk about PFAS today, and I'm going to give you an overview of a recent report that was put out by STAC on PFAS in the Chesapeake Bay, and go through a list of science gaps and potential recommendations to kind of move the PFAS science forward. And Emily's going to talk about how some of these recommendations and gaps are being taken up by the toxic contaminant work group. And then I'm hoping it's going to lead to a really nice discussion here at the end. So PFAS, we've all been hearing about them for quite some time. They're also known as forever chemicals. They've been manufactured since the 40s, as Denise said, by a variety of industries in the United States. They're everywhere in the environment as well as in the human body. And they have the potential to have adverse health effects on both human and critters in aquatic and terrestrial ecosystems. And the fact that there are more than 12,000 unique compounds and probably counting at this point, it makes analysis and reporting very difficult. Most everything these days has or is thought to contain some level of PFAS. We first started hearing about PFAS in aqueous film forming foams, or AFFF, which are used to fight flammable liquid fires at airports, military installations, etc. They're also in your nonstick cookware, your waterproof clothing, paints, etc. And if you take a look at the map on the right, this is source information in those varying blue and brown hexagons. These are from the EPA ECHO database. They're potential PFAS sources, and you can see they're found pretty much everywhere, with a huge concentration along the eastern seaboard. Human exposure to PFAS is really driving public interest as well as state and federal regulations. There are quite a few exposure routes to people, including drinking water, consumption of fish, as well as foodstuffs like crops. For example, PFAS will show up in a wastewater treatment plant. Some of it moves into the waste processing sludge or the biosolids. Then that biosolids can be land applied to agricultural fields, which then can be potentially run off into surface water and or be taken up by crops. While other PFASs make it through treatment and end up in surface water, and some of that surface water is actually where we get our drinking water. And as Denise mentioned, PFAS is quite unique. It affects the immune system. It is not a carcinogen. And regulations have been changing rapidly in the US. It's been very difficult to keep up. A growing number of increasingly strict state and federal water guidelines or benchmarks have been established over the last 20 years. And this timeline is actually pretty quick for regulation. We've seen significant decreases in these guidelines over time, which is primarily due to really improved science on exposure through drinking water and potential human health effects. I do want to note, and Denise mentioned this, there are currently no enforceable national drinking water standards. maximum contaminant levels, or MCLs, for PFAS in the US. There are proposed, which were released on March 14th. However, some states right now do actually regulate for PFAS. The drinking water standards are proposed at 4 nanograms per liter with a maximum contaminant level goal, or MCLG, which means it's a level at which there's no known or anticipated adverse effect of 0. So that means that based on this information that the science is telling us, there's no known safe level of PFAS. But the other piece of all of this, and this is kind of what our STAC workshop or our State of the Science summary got into, is that there's less information available on ecological effects. Because people are driving the regulation, that's kind of where a lot of the work has been put to date. However, we really need to start understanding what's happening from an ecological perspective, because we know that PFAS does affect aquatic species. So in 2022, we had a State of the Science workshop to better understand the state of the science of PFAS, improve science coordination, and really come up with approaches to improve our knowledge of PFAS in the Chesapeake Bay. Specifically, we were focused on understanding and summarizing what's available on sources, occurrence, or presence of PFAS, as well as how it moves through the environment, identify efforts and approaches to really think about these ecological effects on fish and wildlife, as well as trying to understand what states are thinking about new consumption advisories by humans, and then really start to consider study designs and comparable sampling and analysis methods, so we can really coordinate our science across state lines within the Bay watershed. And lastly, the objective was to identify key research needs and data gaps and provide really thoughtful, actionable recommendations associated with a better understanding of the effects of PFAS on fish, wildlife, and their consumption by humans. So prior to the workshop, to try to consolidate as much information as we could, the steering committee created a list of inventory questions that were distributed to regulators and researchers at federal and jurisdictional agencies, as well as interstate commissions, non-government organizations, and academic institutions throughout the Chesapeake Bay. What we were really trying to understand is, what were their priorities with PFAS? What were they doing related to sources and ecological effects? And really where they thought their proposed PFAS work was going to move in the future. From the respondents that we got, we first asked them about sources, because sources, again, we've got point and non-point sources of PFAS. And point sources are like industrial sites, fire training areas, wastewater treatment plants, landfills. Non-point sources would be biosolids, stormwater runoff, etc. So for the sources, the respondents indicated that PFAS programs were really focused on point sources. And you can see the arrow as you move from green to white, that represents most to least studied. So we had a lot of information on point sources from an industrial and a military standpoint, but less on wastewater and landfills. And really, states weren't doing anything with non-point sources. It was more the academic and the federal partners that were focused on those non-point sources. And when we got into short and long-term priorities, they're listed here on the 2 graphs. Again, because human health is the primary driver for studies by jurisdictions, studies on occurrence of PFAS in surface water and groundwater, particularly sources of drinking water, are and will continue to be the priority longer term. There's limited information currently available on ecological effects of PFAS, and these studies are primarily carried out by federal and academic researchers. So we got a workshop together. It was a hybrid workshop. It was one of the first workshops convened by STAC during the pandemic. hybrid in person, which was great because it allowed us to maximize both collaboration and capacity. We had 32 folks in person and 44 virtual attendees from a variety of organizations across the Bay watershed. And we focused the workshop on presentations to really get at the topics listed here on the screen. And then we organized a series of breakout groups to really kind of get down in the weeds and discuss data gaps, needs, and next steps. A number of key takeaways were identified by the workshop participants, and this slide summarizes, I think, some of the most important ones. It is obvious based on the presentations in the workshop that in the responses to the inventory, again, that drinking water and surface water sampling are really the primary focus of the states. However, the issue is that methods are not standardized, which makes it difficult to compare data across state lines and really to kind of think about things more holistically across the Bay watershed. For tissue analysis, PFAS really like to bind to proteins. So if you're interested in ecological exposure and ecological effects, you really need to focus on tissues like blood or liver. But if you're really thinking about people and human consumption, you need to sample the edible portions or the muscle. And lastly, communication and transparency among partners wishing to compare and share data across the Bay watershed is really important. And this is states, federal agencies, academic partners. We need a better framework to really work together and share that information. So from the workshop participants, they also identified 10 high priority science needs that were further bent by the steering committee based on discussions of the workshop by urgency. The urgent gaps focused on more information over time and space on PFAS presence throughout the watershed, including studies that prioritized both fish and water sampling in the same locations to identify individual PFASs of interest and really focus on different sources. Nearish term, so near and near to midterm goals were really focused on ecological and source-based work with an emphasis on effects across different life stages of fish, so from larval minnows all the way up to adults, and under different land use scenarios. So moving away from AFFF and industrial sites to non-point sources of PFAS, particularly in urban or agricultural and urban area or rural areas. Long-term gaps emphasize really more specific questions focused on non-lethal toxicity to oysters and blue crabs, as well as the interface between the aquatic and the terrestrial ecosystem, and trying to put PFAS into context with other stressors like climate change, nutrients, and other contaminants that could affect local fish and wildlife species. So from these recommendations, or from these gaps, a list of 6 actionable recommendations were developed and bent into 3 overarching themes, and they're listed here on the screen. In such a large watershed that encompasses multiple states, communication and collaboration is really essential, as well as designing studies and approaches that will allow for consistent data collection. Developing uniform approaches, including similar methods to standardized data collection, would allow for better comparison more broadly, as well as developing the information needed to better protect aquatic resources. We recognize that additional factors and priorities may exist beyond those identified here. And however, the report reflects a broad effort to bring together knowledge from a range of science disciplines to describe the current state of the science of PFAS in the Chesapeake Bay watershed. Even though the science and policy landscape for PFAS is rapidly evolving and has continued to evolve since the workshop and the release of the report, the science gaps identified are common to many jurisdictions across the Chesapeake Bay watershed. And I think this really presents a unique opportunity to pool resources, streamline methods and approaches, and share findings. And with that, I'm going to hand the screen over to Emily and allow her to kind of give an overview of how the toxic contaminant work group has taken up this report.
Thanks, Kelly. Let me share my screen. Okay, is everybody seeing that okay?
You good, Emily?
Great. So now that we've, Kelly did such a great job of highlighting the science needs and the recommendations from the report. As she said, I'm going to take a couple of minutes to talk about the framework within the Chesapeake Bay Partnership, and specifically within the Toxic Contaminant Work Group. Talk about some lessons learned from other persistent contaminant groups and how we might work towards moving forward on these recommendations in the near term. So just a little bit of background in case you're not aware. There is within the Bay Agreement, as Denise highlighted at the beginning, a goal to ensure that the Bay and its rivers are free of the effects of toxic contaminants on living resources and human health. I find this to be a particularly lofty goal, especially with an expanding scope and without watershed-wide common mandates or regulations like we have for nutrients and sediment. Despite this, we have two outcomes associated with this goal. Those are policy and prevention and research. I'm going to focus today exclusively on the research outcome, as that is where our PFAS activities are associated. The outcome specifically states that we will continually increase our understanding of the impacts and the mitigation options for toxic contaminants through research. And more specifically, the outcome relates to the development of a research agenda that allows us to characterize occurrence, concentrations, sources, and effects of the contaminants, and to identify which best management practices might provide multiple benefits or exclusive benefits to reduce nutrient and sediment pollution, as well as toxic contaminants in waterways. So related to PFAS specifically, I just wanted to show a short timeline that illustrates the prioritization of PFAS within the work group and our research agenda. And you can see that really was first appeared as an emerging issue that we were tracking back in 2018 and 2019. And as our members and work group participants realized the increasing importance of PFAS, we had identified and proposed this STAC workshop. And so what Kelly highlighted was the outcome from holding that workshop, report drafting, and then the release. Recently, in January of 2023, we did move in the revision to our strategy and plans within the partnership adaptive management program. We took PFAS out of an emerging issue and really started to integrate it more holistically into our overall strategy. Going forward, what remains regarding actually implementing some of these recommendations is that there will be a presentation and a solicitation of endorsement of these recommendations to the management board. And we will then anticipate that those endorsed recommendations will be returned to the toxic contaminant work group to integrate them into our existing framework. So what that framework looks like within the umbrella of the partnership is, as I mentioned, we develop a strategy to try to move the needle in the direction of achieving our goal. And, you know, this is guided by a number of management approaches. And these approaches are highlighted in the boxes on the left of this slide. So they include a synthesis of information to make fish and shellfish safer for human consumption, understanding the influence of these contaminants in fish health and mortality, as well as overall distribution of the contaminants in the landscape from different sources, their concentrations and prevalence. And you can see that this framework lends itself nicely to the integration of some of the science needs that Kelly highlighted. So, you know, we would anticipate being able to integrate, you know, our priorities related to fish consumption, fish and wildlife health and effects, as well as impacts of PFAS to ecological life in our streams and different communities in the development of water quality criteria. And then also, you know, with this focus on occurrence and how they're moving and where they may be coming from, as Kelly highlighted, you know, the identification and promotion of consistency regarding design of studies, analysis methods, both field and laboratory, which compounds we're analyzing for, and ultimately how we might store the data are all things that we would be able to incorporate into this existing framework. So the toxic contaminant work group has in the past and continues to spend considerable resources on another persistent contaminant group called polychlorinated biphenyls, even in the research outcome. We can learn from our experiences with PCBs to improve our approach to PFAS as a contaminant class and its presence in the watershed. So both PCBs and PFAS have had bans on legacy contaminants. So we know that there are some PFAS that have been phased out, but they continue to be widespread in the environment long after these bans. Unlike PCBs, other PFAS were developed to replace the banned ones and are still in use today, as Kelly highlighted, in many consumer products, not just firefighting foams. We don't currently understand, as we said, the extent of PFAS entirely in the watershed, but we do not expect there to be a bay-wide TMDL or bay-wide regulatory mandate, just given the experience with other toxic contaminants. This occurs despite the fact that in the example on the right panel, the map showing impairments of PCBs, there are considerable impairments across the watershed. And so, you know, we're faced with having our work group members and jurisdictions facing many similar challenges to a very complex issue. And we recognize that we can all benefit from increased leveraging and coordination, particularly consistency in respect to methods, both field and analytical methods, our assessment of those studies and other topics allow us to make more watershed-wide holistic assessments of PFAS. And in addition, think about long-term restoration over time. Our experience with PCBs is this is a real challenge if at the onset, there are differing methods and approaches. So in addition to the STAC workshop on PFAS, I just wanted to highlight a few additional efforts that the Toxic Contaminant Work Group is doing related to these contaminants. First, the membership has prioritized last year PFAS as a priority, and in so doing, we are dedicating considerable meeting time to the topic in the form of quarterly meetings throughout 2023. These meetings consist of 2 to 3 fairly technical presentations with a question and answer session, followed by a longer working session that allows us to identify what we know, what we don't know, what are the gaps, and what can we do to collectively fill those. We have created a landing page for related resources from the quarterly meetings on the work group website, and we can share that with the group after the roundtable. And finally, as I mentioned, our 2023 update to our strategy and our plan for the research outcome was expanded to include known PFAS studies and monitoring efforts in the watershed. So this includes tracking development of fish consumption advisories in the watershed, ongoing monitoring of both fish and surface water, studies of the effects on fish, and PFAS occurrence with different land use categories. We think that combined with the recommendations from the STAC workshop and the ongoing work, we, as Kelly said, are in a good position to really try to help move the needle in the direction of achieving our goals and doing that in a coordinated and consistent way. So that's the end of my talk, and I'll just, I guess, turn it back over to Denise to start the discussion. Thanks so much.
Oh, thank you, Emily and Kelly. And just a reminder to everyone here, the only way this works is for you to enter your questions in the chat. I have some that were submitted ahead of time, but we'd like to have a robust discussion. So please don't be shy. Let me start off with a couple of maybe more specific ones. The first one is about sources, and I think Emily and Kelly, either of you or both of you can answer this. PFAS and pesticides is a newly discovered source. Is any testing being planned for this?
You want me to take it, Emily?
Sure.
So from what I'm aware with the PFAS in pesticides is that the PFAS are in the containers and then have the potential to leach out into the pesticide mixtures that are then being applied. I'm not sure at this point if there is any work being done on that specifically in the Chesapeake Bay. I do believe that folks are looking at that more broadly, but I would have to kind of do some digging to really understand if there's anything going on in the Chesapeake Bay. I know that there was a talk last year or 2 years ago at pesticides in the Chesapeake, and I believe that a speaker did talk about pesticides and PFAS.
Okay. Thanks a lot. Tom Brewer has a question in the chat. Have there been efforts to categorize PFAS to separate those of concern from those known to be biologically benign?
That's a great question. And the answer is not yet. I think we're still getting a handle on what is actually making it from the source to the water to the animal of interest. We know that PFAS and PFOA are some of the more toxic PFASs, and they are legacy and they have been phased out. However, they are some of the most ubiquitous, and studies have shown that PFAS is taken up readily into tissues.
Great. Emily, anything to add?
No, I don't have anything to add.
Okay. Okay, great. Here's a question on impacts, and I'll intersperse some of the ones that came in beforehand with the ones in the chat. So do we know, is there any sort of ratio, even an approximate ratio or a calculation to really relate PFAS levels in surface water to levels in fish or other wildlife tissue?
Those are called biomagnification or bioaccumulation factors, and I know folks are working on them. Whether or not there is a biomagnification factor for any of the PFAS, I am not sure, but we also know based on studies with PCBs and other persistent organic pollutants that biomagnification factor can depend on species and place in the food web. So my gut is that there are some, but it's just starting to take off again, because a lot of the work on PFAS has been related to drinking water. And so the fact that folks are working on the ecological side of things and starting to understand accumulation is a really important step. In the Chesapeake, there's not anything out there right now. And that was one of the gaps and recommendations, is to kind of combine the tissue and water sampling at the same locations so we could start estimating those accumulation factors.
Mm-hmm. There's a question I'll out of the chat, actually, I'll put onto that, which is a lot of questions on what information do you have on where PFAS enters the aquatic food web? So, for example, is it surface water, pore water, sediment? Emily, that might be one for you.
Well, I do think there are a lot. I think that it is that there are still a lot of questions that we're trying to identify this. And so, I think this particular group of contaminants is unique, that we're sort of in this trajectory of needing to make some decisions, but the science is happening in real time, and we're gaining more information in this exact moment. I think one thing that Kelly said that helps with that and defining, helping us get to that answer of where it's entering, is the multimedia sampling, right? So we've seen with PFAS that sometimes when we analyze the fish, we see particular PFAS compounds that are not in the water, or vice versa. We can also see that they show up in sediment, and the sediment compounds are what are showing up in the fish. And so oftentimes when you have sort of, siloed programs where you're trying to develop a fish advisory, and so you're sampling fish, we lose sight of those other pieces. And so, there is a need, I think, what we're saying is those coupled sampling studies are really important across media. So.
Great, thank you. And Emily, you might want to start with this one first, and
Okay.
Kelly as well, but a question from Chris French. Understanding there are some similarities, but not exact with PCBs, does it make sense to, at least for the short term, align some cleanup efforts that are proven for PCBs with PFAS, especially in regards to possible non-point sources, until we have better data to adjust the cleanup approach? So what might be basically with what you've learned about PCBs, and to the extent that they might behave similarly, is there a short-term strategy while we're gaining information?
So I think I'll start by saying that there was an intentional decision in our workshop to not be inclusive of mitigation and remediation technologies, because that would be a whole other workshop in and of itself. I'm an engineer by training, and so I'm really interested in sort of, gravitate towards the solution. So I appreciate the question. I don't, so just to clarify, so that was not part of the scope of what we covered. I will also say that we haven't expanded our research strategy to be inclusive of PFAS cleanup techniques or technologies at this time. I expect that that will come, but we feel like there are so many gaps on the other pieces of it. We're just not ready to head in that direction. With that said, a full understanding that there is a lot of pressure to act on these. I guess speaking from a PCBs and PFAS perspective, just in terms of their similarities or differences, we at USGS, we have done some work looking at the occurrence of the two contaminants, the two contaminant groups, like within the same study, and thinking about their behavior similarities and differences. So I can't share a paper, a reference with you just yet, but I can say that I personally think that they behave a little bit differently in the environment. And so while there may be some sort of co-location or co-occurrence of approaches, in terms of non-point source, I think that it would be a little bit of a different approach actually than the same. So.
Great, thanks, Emily. That was a great answer. I have a couple of, they're interesting questions about the state of the science on monitoring and analysis. And so let me just sort of ask those. And there are two related questions. One is from Marisa. I'm interested in better understanding how researchers approach PFAS monitoring with accuracy when PFAS are used in so many things, including potentially sampling equipment itself. And there's a related question from Neely Law about the state of the science on monitoring and analysis in terms of being able to detect the presence. And so, for example, is USGS working with local water and wastewater utilities? So there's both, I think, the ability to detect, and then how do you detect when it can, in terms of when you actually do go to sample, there are so many possible ways that you could get cross-contamination. So what do we know at this point about methods for monitoring?
So the USGS is actually working on that. We're in the process of developing field collection protocols for PFAS to get it into our national field manual, which I believe is what some of the states use for sampling. And that's a great point. And so we've had to alter some of the ways we collect samples. We've had to alter the types of bottles we collect in to avoid PFAS. But Joe Duras with the Pennsylvania Water Science Center has been doing a lot on this, not only comparing sampling methods, but also analytical methods. And that was kind of a big piece that we touched on at the workshop, is that there are so many differences in analytical methods at this point until we can actually get folks to use a standardized analytical method with the same number of compounds, the same type of preparation. It's very hard to actually compare the data. So this is a loaded question. And I guess I would kind of conclude by saying it's an issue. And I think the USGS, other federal agencies, and the states are working on this.
Yeah, great. I'm sorry, if I could just add one thing to that. So that was actually the focus of our first quarterly meeting from the TCW in February. And so we had a presentation on the analytical methods and a good discussion about the concerns and variability. as well as field methods that Keller's talking about. So I'll drop a link to that, the materials from that meeting in the chat, so that it might be helpful to some to take a look at.
And what was the second part, Denise, about working with
Well, there
other?
was just about working with, wastewater treatment plants and state water agencies, right? So the question was simply, well, let me see, basically, is USGS working with local water and wastewater utilities in terms of sampling or trying to get to a common method, something like that, and improve methods? Maybe this is related to the question of, I find it so interesting. When you don't have the ability to make a watershed wide, basically standard or TMDL, right, for any of these things, and USGS isn't allowed to like make policy. So you don't have any sticks, and it's even hard to imagine what the carrot is, right? So what have the states, have you gotten any feedback from the states about implementing or how to move on the recommendations that came out of the workshop in terms of common methods and things like that? I mean, is your strategy really to be able to provide the states with a robust sort of regional scale picture of the issues so that they know how they fit within that? Or really, what is your strategy and what's the feedback? Have you gotten any feedback from the states on how they could really move on implementing your recommendations?
So I'm going to let Emily take that on the states moving, because of how we moved that over into the toxic contaminant work group had that discussion. But I do want to note that at the workshop, we had quite a few representatives from the jurisdictions, and they were the ones that were kind of, leading the charge of, we've got to figure out a way to kind of more standardize things, because we're all kind of all over the map. There's nothing uniform. We're all flying by the seat of our pants because, as I mentioned, regulations keep changing. The other thing to note with all of this is that right now there's still no federal regulations for PFAS. There's a proposed drinking water regulation, and it's up for final revision now. And there's also a proposed aquatic life criteria, which leads to potential TMDL development and 303D listing, et cetera. But that's just proposed as well. So, states are kind of grappling with trying to get ahead of things when there's still not even any kind of federal regulations for these. So Emily, can you speak more about the methods, standardization, and things like that with the states through the TCW?
Yeah, so as I mentioned, in early February, we had a conversation about this in our first focused meeting, and I would just reiterate that from what Kelly said, in the sense that it really, the states are being asked to do this, or are actually going out ahead of some of the federal guidelines or recommendations, and I think are really eager to have some input on how to move forward collectively. So they're all sort of having to reinvent the wheel, and so if we can have a forum and a place where these highly technical, complex issues can be, explained, but then also have that conversation of like, how do we how do we sort of coalesce around a certain analytical method, or I think that's sort of organically happening in the way that they're moving ahead, and but they are sort of calling for additional guidance. And so, that's kind of where bringing, expertise from within the watershed, outside of the watershed, making the gap between the scientists and the researchers and then these practitioners that are having to implement, a shorter transition or time, or being able to have that knowledge transfer within the work group. We're trying hard to facilitate that. In terms of actually responding to the recommendations, the report came out, very recently, and we had a very brief conversation on the specific recommendations. So I don't think I have a sense at this moment, a response on those specific recommendations. I would just say that hopefully it was apparent from our presentations, but the STAC workshop outcomes and the trajectory of the TCW are very complementary to one another. They haven't been, different in disagreements on any issues. So, I will expect that there will be, an eagerness to adopt the recommendations that do come back to the work group. So does that answer your question?
Yeah, I think it was a great answer. As this might be a relatively direct one, there has been a lot of requests for is as a result of the workshop or the toxic contaminants work group, or any of this work, has there been a repository identified, like, for example, which agencies are working on the PFAS issue locally? I mean, is there some sort of centralized source of where you're going to deposit, actors and knowledge and all that kind of thing to help each other out?
We hope so. That was
Yeah,
actually part of the recommendations within the workshop, and as from the report, and as Emily mentioned, like, this is pretty fresh off the presses. I mean, this was, I think, just released recently, last month, or earlier this month. So I'm hopeful that we can have a data dashboard or a website or something that can be housed somewhere that kind of compiles all of that information, because, folks at the workshop said it would be great to have that kind of like an expertise board. If you're interested in X, you go talk to, so-and-so.
and my hope is to, with our next quarterly meeting that will be largely focused on fish consumption and fish effects, is that we can start that conversation and, start having the framework at least to get that off the ground. Because particularly with the fish studies at the workshop, there really was a call for, well, if jurisdictions are going out and collecting mussel, there may be researchers who would really like to get samples of plasma or blood or liver from that same fish, and it would allow us to leverage resources and coordinate efforts. And so, my intention was to take the first step in regards to sort of trying to pair those interests and efforts together, but it doesn't exist at this moment.
Thanks. We've had some questions about monitoring and about how to get more monitoring data. So, for example, were there workshop recommendations about, is it relevant at all for citizen monitoring, or is the state of the sampling method so it kind of difficult that wouldn't be a possibility? I mean, what is going on in terms of actual monitoring efforts? And the potential for that. So what's the potential to really, increase the size of the data so that we know a lot more about sources and distribution?
having, I mean, having a volunteer or citizen science network is definitely interesting. It's something to think about and consider. There are definitely pros and cons for a citizen science framework, particularly with something like PFAS, where you have to be clean. You really have to know what you're doing in order not to introduce PFAS into your sampling container. But it can be done. I think the harder piece of all of this is who's going to pay for it. So funding would have to, it would be needed, because these analyses are, hundreds of dollars. So I'm assuming, the states or individual researchers would need an influx of funding to expand monitoring outside of what they're already doing.
Mm-hmm.
But great idea. Let's go find somebody to fund it.
Right. I'll ask you one more question. I just remind people that we will pass everything that we got sent in terms of question that didn't get a chance to get answered. We will send that to the panelists. And when we send you the link to the recording of the webinar, we'll also provide their responses to those questions. So everything will get addressed in one form or the other. But let me ask one last question, and then Kelly and Emily, get ready. I'm going to ask you for just a parting thought, a short parting thought from each of you. But this is a really good question. As if, PFAS wasn't hard enough. Joe DiNardo was asking, is anyone looking at the interactions between PCBs, PFAS, dioxane, heavy metals, et cetera, with respect to health effects, especially since they are all more than likely all in the same fish? And so Emily, I really don't know, from the toxic contaminant work group, I do not know how you get up every morning. But has there been much information on that type of co-occurrence? I mean, it's likely, right? So do we know much about interactions or synergistic or additive effects or anything like that?
So because PFAS that was actually our 10 final science gap. Our last science gap was actually trying to begin to put PFAS in a context with other stressors and other contaminants, because let's be honest, we live in a multi-stressor, multi-contaminant world. And where you have PFAS, you're also going to have other things. So the answer to that right now is no. It's going to be tricky. I think we need a lot more information before we can get to that. But I think recognizing that PFAS is not the only thing out there that affects fish and wildlife is the first step. So kudos. Thank you for bringing that up.
Yeah. I am going to, sorry, I'm going to have to cut it off. Bonnie, see an excellent suggestion about the list of the participants and the chat transcript to perhaps add to the email that goes out with the link to the recording and the answers to the questions. I will let Emily and Kelly have the last word. So if each of you could give us a short parting thought, what you'd maybe like us to keep in mind as we go forward and we're all trying to navigate.
Yeah, I mean, this is a big issue. And there's a lot of moving parts. I think we're just starting to kind of scratch the surface, even though these things have been used since the 40s. So the fact that, everyone is here, everybody is engaged, everyone is asking the really tough questions makes me really hopeful that we will be able to find a solution. It might not be today, it might not be tomorrow, but I'm really hopeful that we'll be able to address a lot of these questions that Emily and I both said, great question, but we don't have an answer for you yet. So keep coming to these things, keep asking these questions, because it's the public that drive regulation and policy and helps fund our science. So please just keep up the good work and stay engaged. And if you're in DC, the 7th through the 13th, there's a ton of stuff happening with amphibians. Come see me. I'll be there.
All right, Emily.
Excellent. Thanks, Denise. And I guess I would just, reiterate what Kelly said, that, that we all recognize that this is an incredibly complex topic and touches many things. And I am also excited at the level of interest and the willingness of parties to come together to really, talk through these challenges and think about the best way in moving forward. So, I think that I would also encourage, come to our quarterly TCW meetings. They are open to the public. I do feel like the best way forward is a collaboration. We really can't keep our blinders on and just look at our jurisdictional boundaries and think about this, thinking about this in a more holistic way. We really all benefit from that. So I really hope that in moving forward, we can take the communication and collaboration recommendation really to heart and start to make some good progress that way. So thanks.
Well, thanks for moving the world in that direction by both of you being here and giving us great presentations and entertaining the discussions. And everyone, thanks for your participation and your questions in the chat. You were wonderfully active, and join us next Wednesday, May 17th, for our next CRC Roundtable. Thank you.